This is the cystic fibrosis transmembrane conductance receptor, CFTR for short. It has two transmembrane domains, seen here in the alpha helices in blue and red. These form an ion channel, which is the main purpose of this protein, and conduct chlorine ions across the cell membrane, which would be located approximately here. The other part of this protein is the nucleotide binding domains, seen down here in purple and yellow. These two are responsible for binding ATP, seen here and over here. Whenever they do bind to ATP, it adopts this closed conformation that you see it currently in. This is whenever it's in its active form, so right now it could be actively channeling ions across the membrane. Whenever these two ATP get hydrolyzed and released from the protein, it adopts what's called its open conformation. What happens then is these two nucleotide binding domains will rotate up and out like this, and then interactions between the nucleotide binding domain and the transmembrane domain will stabilize this open conformation. When it's in the open conformation, it does not conduct chlorine ions. Whenever this protein is non-functional, this is what causes cystic fibrosis. The most common cause of this disease is the deletion of this phenylalanine at position 508. It is highlighted here in green. And what this does is it affects the protein's ability to stabilize its open conformation. So when this nucleotide binding domain rotates up, it cannot properly stabilize this conformation here, which causes the whole protein to become unstable. The cell will immediately tag it for degradation using ubiquitin, usually even before it gets to integrate into the cell membrane.